Hello, we are the University of Lethbridge Collegiate iGEM team, and this year our project is called Club Squared, which provides a method to detect and target a plant disease known as clubroot. As an iGEM team, we are required to tackle local problems and attempt to solve them using synthetic biology. The makeup of our team includes quite a few members involved in agriculture, including children of farmers, students that have worked in the agricultural field, and agriculture majors. This year, the team was given the opportunity to work with an agricultural company. This move made a lot of sense considering the makeup of the team and the resources that could be provided to us. Here is how the presentation will carry forward, starting with the background of our project. Corteva, the agriculture company that we are working with, gave us the option of tackling the problem of Plasmodiophora brassicae, better known as clubroot, which is a pathogen to a variety of cruciferous field crops of brassicae worldwide. Some forms of brassicae include kale, turnip, cabbage, cauliflower, spring greens, and canola. Clubroot is a soil-borne protist pathogen that causes the development of clubs, or galls of varying size on the roots of brassicae crops. These galls result in nutrient deficiency in the plant, eventually resulting in plant death. The resting spore's ability to reinfect crops can occur several years after the last infection occurred.
A major problem caused by clubroot is the massive yield loss it creates due to the formation of galls that inhibit nutrient intake in the rhizosphere plants and hinder their ability to grow. This is a significant problem in Alberta as agriculture is such a prevalent industry. In particular, a common victim of clubroot, canola, is cultivated in our province. Clubroot results in a significant loss of crops and therefore revenue. Additionally, clubroot spores can remain dormant in the soil for up to 20 years and can germinate whenever favorable soil conditions arise. This project is important to our team because we want our farmers in Alberta to be more successful. Clubroot thrives in damp, acidic soil in either cool or warm climates. This is the key reason Alberta may be one of the most affected areas in the world. Clubroot is transferred easily between fields on vehicles and farm equipment, or by wind and water erosion, as well as infection in other crops such as hay and barley, which are commonly used as animal feed. Current mitigation of the disease involves crop rotation to a non-susceptible crop, sterilization of equipment, and using genetically resistant strains of canola. The advantage of crop rotation is that farmers have the freedom of being able to use their fields for crops, just not canola. Sterilization reduces the spread of the pathogen from one field to another via their equipment. Finally, using genetically resistant strains helps as these strains are naturally resistant to infection from clubroot. The disadvantage of crop rotation and sterilization is that they are both timely processes, in the case of years for crop rotation, and that they cause unwanted expenses for farmers. Lastly, using resistant strains requires constant monitoring due to the new forms of pathogen development. Therefore, it is only a matter of time before the pathogen finds a workaround to whatever is causing resistance in the canola.
So we want to emphasize that our team wanted this project to be ambitious but realistic. To do that, we split our project up into two parts, detection and eradication. Creating a successful detection method for clubroot is our number one goal, which is ambitious in itself. But if we can successfully accomplish this step, we would move forward to eradication. Let's see what our detection plan is. Before we do that, an understanding of the life cycle of P. brassicae needs to be explored. So as you can see from the diagram, hour 0 resting zoospores can start to infect the host cells. From hour 12, the primary sporangia forms. Day 1, secondary zoospores start to form. Day 3, the secondary sporangia forms. And then day 14 to 28, the gall forms on canola, forming the clubroot. From day 35, secondary spores can be released and the cycle is repeated. Our project proposes a detection method utilizing an ELISA test kit accessible to farmers, allowing them to easily test their soil from the comfort of their own vehicle. We would like to use the protein PBEL04 as a potential protein target to detect resting spores before the infection of crops. This protein is highly expressed at the early stages of infection and gall formation, allowing for early detection and interference. Our idea is innovative as clubroot does not have its entire genome sequence, therefore it has been difficult to find a specific target in the past. With recent studies, it has been found that PBEL04 is specific to clubroot and expressed heavily during the resting spore phase of its life cycle. Using the protein in ELISA test kits would help farmers know well before they plant canola that their fields are infected. Talking to local agronomists and farmers, they are skeptically optimistic about the idea. At Discovery Seed Labs, they test for the presence of P. brassicae in the soil at approximately $95 a test. We are hoping to produce a product that is not only more convenient, but also cheaper for farmers Canada-wide.
Now we move on to the next step, the eradication of the disease from crops. Our project proposes the creation and utilization of a seed coating containing an engineered bacteria vector, Pedobacter sp, which is symbiotic with canola. Along with this, we will incorporate a cell-penetrating peptide that will target a specific protein, ideally PBEL04, within the clubroot, inhibiting the disease Pedobacter sp is the ideal bacteria to use since it is gram-negative, a prokaryote, already has a symbiotic relationship with brassicate crops, and it has a biohazard level 1, allowing in-lab studies in our facility.
You may be asking why a change needs to be made. To that, we would say we want our farmers to face fewer hurdles. If an increase in crop yield is made, this would then allow for greater farmer production and funds to increase, allowing for a positive effect on the economy. Along with this, it would allow farmers to produce more crop yield, particularly in canola, without having to introduce crop rotation. Here is an overview of the whole Club Squared project. Our aim is to produce a detection method through the ELISA test kit to find the presence of PBEL04. Once this early detection is achieved, the disease can be treated through targeting the specified protein. Both our steps would help farmers greatly, in turn improving the economy. Our first step would be to model PBEL04 and design an antibody for it through the help of our PI. His PhD was in designing antibodies. And then moving towards working with the protein antibody interaction with the help of Corteva.